This is Module 241 of the online course on genetic selection from concepts to application to genetic improvement of health. My name is Jack Dekkers from Iowa State University, and I will be the presenter for this module, in which we will focus on finding genes or genomic regions that affect traits of interest using genomics. In this module, we will discuss the history and promise of molecular genetics applied to understanding complex or quantitative traits. We'll talk about the principle of using genetic markers to identify genomic regions that affect traits of interest by association analysis. We'll talk about strategies for finding genetic markers for genetic improvement and of early examples of genetic tests that have been used in the industry. So to target your learning, I'd like you to keep the following questions in mind as we go through this material. First of all, what is an association study? What is a candidate gene and what's the candidate gene approach? What is a genome-wide association study and how does it differ from the candidate gene approach? What is a Manhattan plot and what genetic markers or genetic tests have been used in the industry? As discussed in previous modules, traditional selection methods for quantitative traits are based on utilizing the phenotype of individuals and of their relatives to estimate breeding values for selection. In this approach, the genetics that underlie the trait is essentially treated as a black box. We know that the trait is heritable and is therefore affected by genetics, but we have no idea how many genes there are, where those genes are, and what their effects are. Nevertheless, as we've seen, this black box approach has been very effective for some traits. However, this approach does require recorded phenotypes on the animal itself and/or on its close relatives, which are sometimes difficult to obtain. And as a result, this approach has limited accuracy for traits that have low heritability, such as litter size, for traits that cannot be recorded on the animal itself, for example because it is sex-limited, or because it requires exposure to disease or slaughter, and for traits that are expensive to record, for example, feed intake. For many decades, the hope has been that these limitations could be overcome if we were able to open the black box of quantitative genetics and then directly select on genes or genetic markers that control the traits. And finally, in the 1970s, some of these hopes appear to become realized with the advent of molecular genetics. So starting in the 1970s, there was enormous enthusiasm in both the scientific community and in the animal breeding industry that new advances in molecular genetics would allow us to identify the genes or QTL that make one pig grow faster than the other and determine where those genes are located and how they function. So then if you can genotype animals for those QTL, you can select the animals that have the favorable genotype at those genes, and you don't have to wait to record phenotypes on the animal and/or its relatives. The problem, however, turned out to be that the location of nearly all QTL was not known, and that tests to genotype animals directly for the QTL could therefore not be developed. What molecular genetics, however, did offer was the ability to genotype animals for so-called genetic markers, anonymous genetic markers, such as the RFLPs, the microsatellites, and now the SNPs that were discussed in an earlier module. Although it's very unlikely that these anonymous genetic markers are the actual causative QTL, some of them may be close to a QTL on the genome and thereby allow the QTL to be identified, at least in terms of their approximate location on the genome. This is done by what's called an association study, which refers to the detection of a statistical association between genotype at the marker and phenotype for the trait. So the idea of an association study is to genotype a group of animals for the genetic marker and then determine whether there's a difference in average phenotype between animals that have alternate genotypes through a statistical analysis as shown here. If the effect of the marker genotype on phenotype is significant, then this genetic marker either is a QTL or, and this would be more likely, the marker is closely located on the genome to a QTL. In this example, you can see how this would occur when animals that have genotype GG for the genetic marker are BIQ for the nearby QTL, and animals that are AA for the marker are LQ for the QTL. So the observed association that we observe between the genotype at the marker and the phenotype for the trait is not because the marker has an effect on phenotype, but because the marker genotype predicts genotype at a QTL, which in turn does affect the phenotype. Because when the genotypes at the two loci are predictive of each other, which is the case here, they are, as we've seen before, they are said to be in linkage disequilibrium, or LD. This is therefore an important concept in association analyses, because without LD between the marker and the QTL, the marker would not show an association with phenotype. And this is illustrated on this slide, which shows a population in which animals that are GG at the marker are not all BIQ at the QTL, but they can also be BIQ or LQ. Similarly, animals that are GA or AA at the marker can have all three genotypes at the QTL. And if the relative frequencies of QTL genotypes are similar for the three marker genotypes, we would expect groups of animals with three marker genotypes to have the same average weight. So although animals that have a different QTL genotype do differ in average weight, this is not picked up when grouping animals based on their marker genotype, because the marker genotype is not predictive of the QTL genotype. It is not in LD with the QTL genotype. So in this case, the marker and the QTL are in linkage equilibrium, which can happen in a population, as we've seen, even for loci that are linked. And so genotyping a marker that is linked to a QTL does not guarantee that that marker will allow you to capture the effect of that QTL on phenotype. The marker and the QTL also need to be in linkage disequilibrium, not necessarily complete disequilibrium, as in the previous slide, but they have to be at least partially in linkage disequilibrium. But the effect that you see at the marker will be greater or greatest if the LD between the marker and the QTL is high, is complete, and you'll see the full effect of the QTL at the marker if they are in complete LD. This is a graph that we discussed in a previous module on the relationship between LD and distance between pairs of loci. This shows that only loci that are closely linked can show substantial LD. Thus, if we want to find markers that are associated with phenotype, we need to focus on markers that are close to the QTL. However, not all markers that are close to the QTL will show an effect on phenotype, because not all loci that are close together are in high LD, as indicated also in this graph. And that leads to two approaches for deciding which markers should be genotyped for association studies to identify the QTL. One is the candidate gene approach, and the other is the high-density SNP genotyping approach. Now, if you have limited funds for genotyping, you could take a targeted approach in which you genotype animals only for markers that are in a region that you think harbors a QTL. And this prior information could be based on the biological role of a gene in that region. It could be based on a previously detected QTL in another population. It could be based on comparative data from other species, or it could be from gene expression data. For example, if you have found that a particular gene increases in expression if the animal is exposed to disease, then that would be a good region to investigate markers for their association with disease susceptibility. One of the first successful applications of the candidate gene approach in pigs was by my colleague Max Rothschild at Iowa State University in the early 1990s, and he used the estrogen receptor gene as a candidate gene for litter size because of the role of estrogen in reproduction, fertility. So he genotyped pigs for several RFLP markers in and around this gene, and he found one marker that had a sizable association with litter size. This was then repeated in larger numbers of litters, and the results showed, as shown here, that one copy of the B allele at this marker was associated with an increase of almost half a piglet in litter size in both first parity and also in later parities. They were subsequently able to show that this genetic marker was itself not a causative gene, since it occurred in a non-coding region of the gene. So this is an example of a marker that is associated with phenotype as a result of it being in LD with a nearby causative mutation. Now, interestingly, that causative mutation has not yet been found. But nevertheless, this marker was used by PIC in several of their lines to improve those lines for litter size. Another example of a successful candidate gene, also by my colleague Max Rothschild, is the MC4R gene. MC4R was selected as a candidate gene for growth based on results in mice, which showed that a knockout of this gene resulted in a substantial increase in growth rate, as shown here, where the mouse on the left is a knockout and on the right is its full SIB without the knockout. With this knowledge, Max and his PhD student, Kwon-Suk Kim, set out to look for genetic polymorphisms in the MC4R gene in pigs. And they found one point mutation in a coding region of the gene that changed one amino acid in the transmembrane domain of the receptor protein that is produced by MC4R. And then they developed a genetic test for this SNP, and after genotyping large numbers of pigs for this mutation, they showed that genotype at this mutation has a substantial association with back fat, loin depth, daily gain, and also feed intake. And through functional analyses, they also were able to show that this indeed was a causative mutation. Although the candidate gene approach has led to some successful tests, generally these tests only explain a limited amount of genetic variation for the traits. The main reason for it is that it's a very targeted approach, and it covers only a small proportion of the genome. This limitation was, however, overcome by substantial developments genotyping technology in terms of high-density SNP genotyping. And that allowed animals to be genotyped for large numbers of SNPs across the genome, so it's that each QTL, wherever it is on the genome, has several SNPs close enough for at least one of them to be in sufficient LD with that QTL to show an association with phenotype. This revolution started in 2000 with publication of the first draft sequence of the human genome. In 2004, this was followed by the release of the first livestock genome, which was that of the chicken, and then genome builds for cattle and pigs followed soon after. Although having a template of the complete genome of animals that we work on is useful, what was even more useful was that the sequencing technology that was developed allowed us to determine where genetic variation or polymorphisms occurred across the genome. For example, soon after the release of the chicken genome, this sequencing information was used to identify nearly 3 million places along the genome where breeds of chicken differed from each other, and many of these places were also segregating within breeds. And similar libraries of SNPs were developed in other species, which now gave us access to many more genetic markers across the genome than were available before. However, there was one other advancement that was needed for this to be useful for animal breeding, and that was the high-throughput technology to be able to genotype large numbers of animals for this large number of SNPs across the genome. And this became available in cattle in 2009 in the form of the 50K Illumina SNP chip, which allowed one individual to be genotyped for 50,000 genetic markers across the genome for about $200 initially, which now has dropped to less than $50 per individual. Now, in comparison, in 2000, the cost to genotype one animal for one genetic marker was close to $1. So in 10 years, genotyping costs dropped from $1 per data point to less than half a penny per data point. And similar SNP chips and SNP chips with over 500,000 SNPs are now available for most of the livestock species. So these tools allow us to genotype substantial numbers of animals for large numbers of genetic markers across the genome, and this increases the chance that a QTL, wherever it is on the genome, has at least one SNP near it in high LD. Now, to detect these QTL and SNPs, we conduct an association study for each SNP, one at a time, to detect associations between marker genotypes and phenotype. Because this is done for each genotype SNP across the genome, This is referred to as the Genome-Wide Association Study, or a GWAS. In this analysis, we can either fit the marker genotype as a class variable, such that we get least square means for each of the 3 SNP genotypes, or we can fit SNP genotype as a covariate based on the number of 1 alleles, such that we estimate an allele substitution effect or an additive effect. And tests of significance for the genotype effect then give a p-value for each SNP, which we can transform by taking the negative of the log 10 of the p-value, such that lower and thus more significant p-values result in higher transformed values. So a p-value of 0.1 becomes 1, and a p-value of 0.001 becomes 3. The resulting minus log 10 p-values can then be plotted for all SNPs across the genome in what's referred to as a Manhattan plot, because it resembles the skyline of Manhattan. So in a Manhattan plot, each dot represents the association analysis result in terms of its minus log 10 p-value for one SNP, with higher values indicating greater statistical significance of the association of that SNP with phenotype. Now this graph clearly shows several regions where multiple SNPs are associated with phenotype, indicating that there's a QTL in that region, and that multiple SNPs in that region are in LD with that QTL. The red horizontal line shows the significance threshold, which is fairly high, 10 to the negative 7 in terms of a p-value, because it accounts for multiple testing. Now, one of the problems with these genome-wide association studies is that the statistical power typically is limited, especially because of the large number of tests that are being conducted, and therefore we only detect QTL with large effects, which is a limitation. Nevertheless, these high-density SNP chips were used to develop small panels of SNPs that were associated with traits of interest and used for genetic selection in pigs, in particular by PIC. And this figure shows the timeline for the use of genetic markers by PIC as an example of one breeding company. It started with the Haldane test, or the stress gene, followed by several markers that were identified by the candidate gene approach, such as ESR, the estrogen receptor gene, the MC4R gene, and the RN gene. Other single genetic tests that were developed and used were the FUT1 gene, which blocked an E. coli receptor in the gut, preventing diarrhea. And a very interesting genetic marker was for a mutation in the IGF2 gene, which was found to be associated with lean yield, but only if the lean allele was received from the sire rather than from the dam. And this is an example of what's called a paternally imprinted gene, meaning that only the allele that is received from the father is expressed in the offspring. Now, this offered some unique breeding opportunities by mating a homozygous lean boar to a homozygous fat sow to produce heterozygous market hogs. Now, the uniqueness of this strategy for this paternally imprinted gene is that it produces market hogs that are lean, even though they're heterozygous, because only the allele received from the sire is expressed, which is the lean allele. However, the sow that produces these piglets is homozygous for the fat allele, which has some advantages for her, because she has then the reserves to sustain pregnancy and lactation. So through these candidate gene and GWAS approaches, PIC was able to increase the number of markers that they used in their breeding program from just a couple of markers in 1994 to about 100 in 2001 and about 157 in 2010, with markers for a range of traits, including disease resistance. So this concludes this module in which I've introduced some of the approaches that were used to identify genetic markers and genomic regions that are associated with phenotype, for traits. Now, to check your knowledge, please try to answer the following questions. What is an association study? What is a genome-wide association study? What is a candidate gene and what is a candidate gene approach? What is a Manhattan plot and what is it used for? And what genetic markers or genetic tests have been used in the swine industry in the early years of the adoption of molecular genetics? Thank you for your attention.